Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer in the comment below, so you can also answer question to each other for solving the problem. So today we will discuss about the introduction to cell signal. That is the meaning of cell. That's a one cell is signalized to another for specific function. On the other hand, the intracellular communication, that is cyclic AMP second messenger, we will discuss about in detail. So here first of all we will discuss about the overview of the different cell signals by a different mechanism. So first of all, I want to say that is the ligands. The ligands is the hydrophilic, lipophobic, lipophilic, or hydrophobic ligands. That the ligands is the gases, can be fats or protein and sugar. So here the ligand will ligand are present in the cell, which stimulate to another cell by a receptor. That receptor is for example paracrine hormones. So the paracrine hormones mean the one cell which that stimulate to another cell for the for example muscle contraction of the heart, which that is the ultimately all muscle cell will be contract of the cardiac cell. So that's the one cell stimulate to all cell is known as the paracrine hormones or paracrine system. That's a paracrine stimulation. So that is the produced by the receptor of the ligand binding to that create the cascade of different mechanism. That means that is the production of something. So the hydrophilic, lipophilic, lipophobic and hydrophobic, as well as basically that's ligands. And ligands can be gases, fats, protein and sugar. So that's all is a stimulating agents which stimulate the cell to further produce a cascade of different mechanisms. So important to know that here we will discuss about the with a briefly overview of that is the basically here is the receptor, and the receptor can be made of the protein. That's a protein receptor, and the ligands is bind with the receptor. And when ligands will bind with the receptor, and that is here is the plasma membrane, so that is known as the reception. So the reception means the receiving of something from the ligands, for example fat, lipoprotein, etc. So that is also that is a receptor. So that's activate and produce a cascade of different mechanism to bind with the DNA to produce RNA, and that is known as the transcription. And the transcription of the messenger RNA to produce protein by the different mechanism. No, here we will not discuss about the mechanism of the protein synthesis. So here is the reception, and here is the transduction. So the transduction means the activation of the precursor of molecule which that is bind with the DNA to give a response. So that is here is the response. Anyhow, here we will discuss about the all ligands or ligands. The ligands, that's ligands will be bind with many different mechanisms to stimulate and activate the cell activity. So here I am drawing the synaptic ligands. That's a synaptic ligands is attached with a neuron to neuron is known as the synapse, and this synapse is the production of acetylcholine neurotransmitter which that release and bind with the receptor of the postsynaptic neuron, and the presynaptic neuron and postsynaptic neuron which that stimulate to increase the nerve impulse, that is the action potential. So the generation of action potential is due to the binding of the ligands is also is the binding by the neuron is the stimulate by the action potential due to the changing of the ions due to the channels, activate the channels. On the other hand, here is the intracellular. Intracellular response mean the ligand will go into the directly into the cell, and that's directly into the cell is, for example, steroid, which that is a testosterone or progesterone, estrogen, and cortisol hormone, which that is bind with the protein receptor. That is a protein, and ligand will be bind and go into the receptor of the DNA to give a response. On the other hand, that is intercellular signal. So the intercellular signal mean the two cell will be ligand will be bind with the receptor to stimulate the cascade of different mechanism to produce a protein is the signaling which that is the signaling protein which that is bind with the receptor to produce a mechanism. So that is a paracrine signal. On the other hand, that's paracrine signal, which that is mean the cell to cell mediated response. So here I am drawing that is the for example, that is the major overview of that diagram, which show that the, for example, the neuron will be bind to give a signal by the acetylcholine as a acetylcholine receptor, which that is play as a important role as a ligand gated ion channel. So the ligand gated ion channel will open when ligand will be bind with the side of the channel. So that's here is the basic receptor we receive, and that's activated. That's against concentration gradient to produce by the using of ATP to incoming or outcoming of the sodium, potassium, and chloride ions, which that is, for example, here is the hypothalamus, which there is a neurosecretory cells contain a neuron which that secrete the acetylcholine neurotransmitter, and here is the acetylcholine receptor by the activate of the pituitary cell to produce or release the hormone. So that is the ligand gated ion channel is here. So on the other hand, here I will discuss that is a nervous signal. Nervous signal mean that is the primary signal is from the neuron, that is the synaptic vesicle will be released as a ligand, and that's bind with the receptor of the nervous ligands, that is the neuron attached to the cell. For example, in the pituitary gland, that is secrete the vesicle by the activation of the cell, and that's ligand will activate to release the vesicle, for example, protein or hormone, etc., which that's go into the blood vessel, and that's produce that's go into the blood vessel, and that's bind to another cell to release, for example, thyroid stimulating hormone, which that is produced from the pituitary. So that is go into the blood to release. So that is known as the endocrine signals. So the endocrine signal, for example, the gonads. On the other hand, here is the basically pituitary gland, for example, the cell of pituitary gland. So next we will discuss about the autocrine signal. Is autocrine signal mean the cell which that is use the itself the stimulation. For example, sometime the blood will be produced. For example, the blood will be allowed to enter the ligands, which that's enter into the blood, and that's entry of the blood which circulate throughout the body, and when the need of the secretion of something, that's a ligand will be bound with a receptor to release into the blood. So that's releasing mechanism will be produced, and that's production of that is known as the autocrine signal. Auto mean itself, and crine mean that's a production of the hormone. So now we will discuss about the whole mechanism of the intracellular communication and cyclic AMP second messenger as a second messenger. So the second messenger mean, here we will discuss about the second messenger is the cyclic AMP. So the cyclic AMP means the cyclic like cycle like structure which that is bind, and adenine monophosphate will be formed. So here is the pathogen cells mediator signal which that is, for example, HIV will be bind with the CD4 receptor of the helper T cell to disrupt. So that is the disease which that the ligands are present in the HIV virus, which that is sometime HIV will be bind with the CD4 receptor to enter into the enter into the T cells. So that T cell will be damaged by this mechanism. On the other hand, here we will discuss about the cell. So the cell which that is that's giving the signal, how to produce the signal, and how to express the gene from this signal is we will discuss with this detail. So here is the extracellular fluid of the cell, and inside is the cytosol. So for example, there's a messenger. There, for example, the messenger is the ligand lipophobic. So the lipophobic means that that's ligand can't be entered into the cytosol without any without any changes. So that's directly not directly move inside, but the cascade of different proteins used to stimulate that second messenger. So here is the messenger ligand lipophobic. That is the primary messenger. When primary messenger will be bind, that's the primary messenger which is the, for example, some hormone, for example, growth hormone or some hormone which that's bind with the here is the G protein. So the G protein here is the receptor. That receptor receive the ligands which that is the lipophobic and not interact with the basically with the plasma membrane. So here is the G protein, which that is a G protein is made up of 3 subunits. So the 3 subunits contain alpha, beta, and gamma. So the alpha subunit contain a GDP. GDP means the guanosine diphosphate. So this guanosine diphosphate will be converted by the GTP to convert GDP, and that's give that phosphate group that's produce the activation. So that number 2 is the activation of the G protein by the using of ATP. Is that is the mechanism we will discuss. So the ATP is used to give the phosphate group to the GDP, and that GTP convert into the GTP. And here is we will discuss about where the ATP is formed. So here when insulin receptor will be bind from here, there's an insulin receptor. So that's insulin receptor site, and that's open the GLUT2 channel. So that's GLUT2 channel, for example, open to increase the fructose and glucose entry into the cytosol, and the glucose will be enter, and that's glycolysis will be formed, and this glycolysis is produce the pyruvate. That's 3 carbon containing molecule, and there's 3 carbon containing molecule will go into the mitochondria to produce acetyl-CoA. And by the using of oxygen, the acetyl-CoA will be go into the Krebs cycle with the oxaloacetate to bind with acetyl-CoA to produce alpha ketoglutarate, etc., which that is produce ATP. And this ATP and carbon dioxide will be released, and that's ATP will be produced from that mechanism. So next here we will discuss about the GDP will be converted into the GTP, and that's alpha subunit will be released, and that's bind with the adenylate cyclase enzyme. So that's adenylate cyclase enzyme, which that is the amplifier enzyme, which that's amplify, and that's ATP used to make a cyclic AMP, and 2 phosphate group will be separate. So this GTP guanosine triphosphate will be bind with the alpha unit of the G protein. So the G protein contain 3 subunits. So here is the ATP will be bind, and that's ATP produce that cyclic AMP, and this cyclic AMP is the second messenger. So the second messenger is mean the first messenger was the ligand, which that was the lipophobic. On the other hand, the cyclic AMP is the second messenger, which that means it's activate. How to activate? That activation is known as the protein kinase A, which that is activate the protein kinase A, and this protein kinase A will activate the protein bind with the phosphate group, and this protein binding with the phosphate group is through that mechanism. For example, the protein will be bind with the ATP, and this ATP used to produce ADP, and phosphate group will be transferred to the protein, and that protein is called the protein phosphated protein as phosphorylase protein, and this protein is the mechanism, and this protein is something. For example, different type of protein will be formed, and that's here here is the same mechanism, but the primary messenger is the ligand lipophobic, which that is the different. For example, the different hormone which that is bind with the receptor to produce this mechanism, and that's produce the second messenger, and this second messenger will be formed, and here is the protein will be formed, phosphate containing group. So important to know that the ligand will be released. After the releasing of the ligand, there's a mechanism will be produced. Here is the phosphorylase protein will be bind with the receptor. So that the receptor is present in the DNA, and there's a polymerase enzyme used to create the transcription, and this transcription produce messenger RNA. So first of all, we need to know about that the primary messenger will be released. So here the transcription will be formed. So next we will discuss about the releasing mechanism of the primary messenger. So important to know that here is the important things. Here is the transcription factor will be formed. This transcription factor is the special and unique. So this unique transcription factor will bind with a specific segment of the DNA for translation of different protein. For example, muscle protein. For example, liver cell protein. Liver produce some protein or antibodies. Different type of protein will be released. For example, immune system will be produce the protein, or complementary system will produce a protein. So here is the primary messenger is the ligand lipophobic will be released. When releasing mechanism will be backward with the G protein back to the alpha subunit of the protein, which that is again bind with the receptor for further signal. So here is important to know that here will be we will discuss about that is the cyclic AMP will be reversed. For example, the basically here is the phosphodiesterase is the enzyme which that is the PDE, which that is used for the breakdown of the second messenger. So the second messenger breaking down to release as make a ADP, and that's ADP ultimately convert into the ATP from the mitochondria. So here's the ATP used to produce ADP, and that phosphate group will be given to the cyclic AMP to convert ADP, and this ADP will be converted into the ATP, and this ATP will be come from the mitochondria. For example, ADP will go into the mitochondria for further production of ATP. So the next is the protein protein phosphorylase protein is here, which that is break down by the dephosphatase the protein, the dephosphatase protein, which that is the enzyme used to break down, and this breakdown is the backward to release the protein plus ATP, and that's protein as release from it. So that is the backward mechanism. So produced from the backward. So this protein will be released and ATP will be separate. How it's possible? Because ADP will be add with the phosphate group, which that was the add into the protein. So here is the messenger RNA will be produced for further process. So that is the translation to produce protein. So this protein is the specific. For the specific is known as the phenotype. So there's a DNA contain a genotype. So that genotype will express the phenotype of that things. For example, production of hormone, production of something. For example, production of protein for synthesis, protein for the muscle, myocyte, cardiac cell or something. So thanks for watching. Please subscribe the channel. Okay, bye.